Hello, my name is Connor Nason, and I will be discussing protein synthesis. So what I want to bring up first is central dogma, which is deoxyribose nucleic acid being transcribed as messenger ribose nucleic acid, and the information from that is translated into a protein. So in transcription, DNA serves as the template for making messenger RNA, and so that means it'll be having a sugar, a phosphate group, and a nitrogenous base, but it will not have the nitrogenous base thymine that is only found in DNA. There are three types of RNA produced during transcription, which are messenger RNA, transfer RNA, and ribosomal RNA. There are also three events that occur in transcription, which in the initiation of transcription, DNA is unwound by enzymes, and then an enzyme called an RNA polymerase, which synthesizes mRNA copies. It attaches it to a DNA strand at the promoter region or start point, and the strand that is copied is the template, and the one that is not is the coding region. In the elongation of transcription, free nucleotides will be paired, and the RNA polymerase will move down the strand, reading and pairing, and once it'll stop once it's finished or until it reaches a stop signal. So, and then in the termination of transcription is where RNA polymerase reaches a stop signal. It is released and DNA will rewind into a double helix. And whenever initially synthesized, mRNA is known as pre-mRNA, so it needs to go through a process called splicing so it can mature and become mature mRNA. So in translation, the information from mRNA is used to synthesize a protein. So this will occur at the ribosomes within the cytoplasm, and the information of mRNA is translated from mRNA and into an amino acid. So in the initiation of translation, methionine is bound to tRNA, and the start codon will be on the ribosomal P site. And in the elongation of translation, the subsequent amino acids are delivered, and then the peptide bonds will start to form, and the whole process will repeat until the whole mRNA sequence has been translated. So in the termination of translation, a stop codon will enter the A site and it'll end the translation, or until a new protein is formed. That is also whenever the translation ends. So some protein synthesis disorders are usually caused by errors in the synthesis of proteins, and this would cause misfolded proteins or insufficient protein function, and Alzheimer's, Parkinson's, cystic fibrosis all result from the genetic mutations that change or affect tRNAs, rRNAs, and ribosomal proteins.